Hello, we are Hyun and Hyun in the Neuroscience Technology class. In this video, we are going to present about imaging brain activity with multiphoton excitation. The contents include non-degenerative photon microscopy, three-photon microscopy, wearable multiphoton microscopes, and adaptive optics in multiphoton microscopy. Part 1 is about non-degenerative photon microscopy. Two-photon microscopy is a type of fluorescence microscopy. In two-photon microscopy, a single photon cannot excite a fluorophore because each photon has twice the wavelength and thereby half the energy required for excitation. Therefore, a fluorophore can be excited by observing two photons simultaneously, which is an extremely rare event. So, fluorescence excitation is restricted to a narrow plane of focus within a specimen. Two-photon microscopy has some important advantages over one-photon microscopy. First, depth limit for imaging is much deeper, because photons in two-photon microscopy have wavelength twice as long as photons in one-photon microscopy, so the photons penetrate deeper into the tissue. Also, because excitation of fluorophore is restricted to a focal point, two-photon microscopy causes much less photobleaching and phototoxicity. However, there are some limitations of two-photon microscopy. First, two-photon excitation is inherently weaker than one-photon excitation, leading to darker fluorescence. This problem can be solved by using non-degenerative photon microscopy. Second, the depth limit of two-photon microscopy is still too shallow to image subcortical structures. Deeper imaging can be performed by three-photon microscopy. In degenerative photon excitation, fluorophores are excited by absorption of two photons of the same energy. However, in non-degenerative photon excitation, two photons of different energy are simultaneously absorbed. In non-degenerative photon microscopy, or ND2PM, two photons of different color are derived from two laser beams that overlap only in the focal plane. The advantage of ND2PM over D2PM is that signal-to-background ratio is improved. First, in D2PM, excitation occurs not only in the focal volume, but also near the surface, which is called out-of-focus excitation. However, in ND2PM, excitation occurs only in the focal volume. Also, the probability of two-photon excitation is higher in ND2PM compared to D2PM. Therefore, ND2PM provides improved quality of an image, even for the deeper penetration depth. Part 2 is about three-photon microscopy. In three-photon microscopy, each photon has one-third of the energy required for excitation of the fluorophore, so a fluorophore can be excited by absorbing three photons simultaneously. Three-photon microscopy allows deeper penetration than two-photon microscopy because wavelength of photons is longer. Using the deeper penetration of three-photon microscopy, non-invasive in vivo imaging of subcortical structures within an intact mouse brain became possible. Figures in this slide shows the reconstructed three-photon microscopy images of mouse brain vasculature and neurons. Three-photon microscopy provides clear image even in the penetration depth of deeper than 1000 micrometers.
The part 3 is about wearable multiphoton microscopes. Miniaturized microscopes enable imaging in freely behaving animals. So there have been continuous attempts to miniaturize multiphoton microscopes. Even they get smaller, advantages of multiphoton microscopes compared to one-photon microscopes are maintained, so they offer laterally and axially higher resolution and larger depth penetration. Also, as you can see at the second figure right down side, miniaturized two-photon microscopes have similar resolution and contrast with benchtop two-photon microscope. But those miniaturized microscopes also have limitations. They have low frame rate and smaller field of view, which complicate stable recording. And also they need low repetition rate amplified laser system like the short pulse laser and special fibers like hollow-core photonic crystal fibers that deliver light in low dispersion, and photomultiplier tubes that detect weak light signal by multiplying it. On the right side is the schematics of two-photon and three-photon miniaturized microscopes. And the last part is about the adaptive optics. During brain imaging, there could be wavefront distortions which aberrate the focus, but adaptive optics methods can reduce blurring effect of wavefront distortions, aberrations, and diffraction. AO system control and correct excitation light distorted wavefront with a phase device such as the deformable mirror or the wicked crystal spatial light modulator located between the objective lens and the beam splitter. So, with AO system, in morphological imaging you can get better resolution and contrast like the pictures below that show the brains of mice, flies, and zebrafish respectively from the top. Also, AO system can be adaptive in functional imaging too. With adaptive optics, brightness and contrast of imagery are increased, and amplitude of calcium transients increases. Also, the signal noise ratio increases too, because of reduced excitation focal volume and enhanced focal brightness. Then, the calcium transient and tuning curve get sharpened, as you can see in this figure. So, we can say that adaptive optics is almost essential for tuning curve characterization. These are the references, and thank you for listening.